So I'm going to talk to you about some of the work we've been doing in Adalta to try and address fibrosis. And we'll be talking about the lead compound, AD114. I'll explain what that is. You know, one thing we've learned from being in this space is that a lot of people outside there in the general public don't really understand what fibrosis is. Some of them have never even heard of it, except in the context of cystic fibrosis. But, and I'm not going to go into it because there's a bunch of experts who are going to tell you a lot more about it. But it is a severe disease, and it's actually prevalent in the end result of a lot of different types of diseases. Fibrosis can occur in lots of different tissues, and we'll discuss a couple here. And our initial focus is on lung fibrosis, but we have some really interesting data in the eye and in some other tissues. So what is an eye body? An eye body is a very small antibody. So this is a human antibody. This is what we have in our blood. This is the antibodies that are very popular as drugs today. So they're replacing a lot of the smaller molecules that you heard about earlier. And that's because they've got incredible affinity and incredible specificity. So they tend to have lower side effects. This is an antibody that's in the shark. And you can see both of those molecules are large, complex, at least in the molecular terms. But the shark antibody has evolved to basically act as a single domain. And so what we've done is to take the single domain. We did a lot of work. We showed that you could actually use this in the laboratory to target different diseases. But the problem with a shark antibody is it's a foreign protein. And so if you put it into your blood, it's going to be rejected. And so we've developed an eye body, which is essentially a human equivalent. We just did some molecular tricks, but it has the properties, or similar properties to the shark. And that means it's got high affinity in general, it's got high specificity, and because it's got a big long loop, it seems to be able to get into areas of proteins that traditional antibodies can't get to. Sorry? Oh, I thought I heard somebody. So our lead candidate, 114, I think I've largely said a lot of this, but we've demonstrated the antifibrotic activity and anti-inflammatory activity of this compound. So if you're going to target a disease, you need to find a target molecule or a target protein that's involved in the disease. And our target is CXCR4, so the name's not important, but this molecule has been shown quite clearly to be involved in different types of fibrosis. And this paper here showed that if you have these fibrocytes, this cell type in your body which expresses this protein, then if you have more than 5% fibrocytes, your chances of survival are about 7.5 months. If you have less than 5% fibrocytes, you can live for 27 months. So it's still pretty desperate, but I think it does implicate this protein in the progression of fibrosis. And so we sent our eye body to colleagues in the US who looked on patients, and he split them up into patients that progress slowly and patients that progress rapidly. And our eye body was able to bind CXCR4, the target molecule, was upregulated in those cells from those patients. So in collaboration with the guys in the US and colleagues at the Alfred, we've shown quite clearly that AD114 doesn't bind to normal lung tissue. So this is normal lung tissue from non-disease people. And this is Adalta AD114 binding to lung tissue from people with fibrosis. So you can, I don't know if you can see it, but the brown staining there is the binding of our eye body to cells in that in the tissue from lung fibrotic patients. What we then did was take those fibrosis tissue samples and showed that the eye body, for example, would be able to slow down the rate of migration. So how fast these cells move. This is just a time course showing you that if you don't have any drug there, they migrate at this rate. When you add the 114, the eye body, shown in the red here, you can see that it's depressed. And so it significantly reduces the migration of these cells. Importantly, our eye body doesn't seem to affect the normal fibroblasts from normal tissue. And so I think that was quite important, and it differentiated us from some of the other CXCR4 drugs. There's a small molecule, CXCR4 drug, and like Robert mentioned today, this has got severe side effects. It has got cardiac toxicity. And then there's the two other drugs, nintedanib and pirfenidone, which have just been registered in the last few years or given to patients in the last couple of years. And I think it's fair to say that these are, they're not stellar, these drugs. And they have different profiles to our eye body. So our eye body is able to affect both fast and slow progressors, but not normal fibroblasts. So that's great. You do it in the laboratory, but will it work in an animal? And so we tested our eye body in the bleomycin model. So bleomycin is just a toxin that if you give it to mice in the lungs, you form a fibrotic-like disease. So animal models, they're not perfect, but this one is the gold standard, and it's what the FDA want you to do. So you can see lung tissue from normal mice, nice airways. When you give the mice bleomycin, you get massive deposition of collagen, and this is the stiffening of the lungs that happens in fibrosis. If you give the mice bleomycin and at the same time dose them with our eye body, then you really prevent that deposition of collagen and influx of inflammatory cells. And this is just a quantitation of that, so you don't need to worry. I think the picture speaks pretty clearly. This was done in what we call a prophylactic mode or a preventative mode, where we give bleomycin and we give our eye body at the same time. And that's not very realistic when it comes to therapy. You can't predict who's going to get fibrosis. So the FDA would like to see where you do it you induce the disease, and then at some point later you give the eye body. And so while these data are not quite as clean as the other one, because you've started fibrosis, I think it's clear that the normal tissue, IPF lung tissue with the bleomycin, and when you add 114, you're slowing the progression of that of the disease. So this was the way we did it. Again, we quantitated it, and that was satisfactory for the FDA to give us orphan drug designation in this space. We've also said, so if the eye body can work in lung fibrosis, can it work in other types of fibrosis, other tissues? So we look at NASH in the liver, and we've shown that the eye body has got antifibrotic effects in the NASH model. This model again is not the best model, and I don't know if you can see this, but this is normal liver tissue. NASH is a model of liver fibrosis. And so again, in a similar way to the bleomycin model, you induce fibrosis, then you add the eye body and see what you can affect. And while it doesn't go back to normal, it's definitely there's less damage to the liver. And the quantitation shows that the eye body has essentially dropped everything. Some of them don't reach quite statistical significance, but it drops it all. And most importantly, I think this score here, it's called the ballooning score, shows clearly that there's protective effects of the eye body in the liver. What about fibrosis in the eye? We're collaborating with a colleague at Melbourne University, Erica Fletcher, and AMD, of course, is a very important disease, and there's a fibrotic component, so there's fibrosis involved in macular degeneration. And again, we've tested our eye bodies in this space. So here what Erica does is to laser the eyes of a mouse. That induces leakage, blood leakage, so it mimics the AMD situation, the macular degeneration situation, and then added our drug intravitreally, so injected directly into the eye. And that's what happens if you get AMD and you go to get treatment, you'll get injection in the eye. And what she saw was that when you treat with a laser, you get this quite dramatic contraction of the retina. when our eye-body was added at the same time, you basically blocked that contraction. And this is the quantitation here. It did affect leakage size, not quite as dramatically as it affected that. And this here shows that the eye-body was responsible for dampening a whole bunch of genes that are all induce fibrosis. So it seems to have a broad effect on a lot of different profibrotic genes. This is a model in the, and so again, that was in the preventative mode where you give the eye-body at the same time as the damage. This is a more realistic situation where you give the damage, then 3 days later you give the eye-body, and then you look 4 days after that. And I think it's clear, again, the eye-body has a little effect on leakage, but not very much, and certainly not as good as Eylea, which is a drug in the market at the moment. But the fibrosis is dramatically reduced with the eye-body. And it's sort of about the same as Eylea at much lower doses. And so,
so I'm guessing you haven't done the work yet, but presumably it would be synergistic with Eylea. We'd probably find it was synergistic with Eylea.
It could be, because they're both, yeah, okay, we haven't done the work, but they're both acting through different mechanisms. One of the problems with Eylea is AMD, you get macular degeneration. Eylea essentially stops the blood from getting there. It's a VEGF inhibitor. It stops it. So what you end up with is dry AMD. So you actually get atrophy of the eye if you're on Eylea for too long. So it might be good to try and either see if they both work synergistically, or if the eye-body works alone and stops the fibrosis. So I think, just to summarize, you know, our technology, these eye-bodies, we've taken the lessons from the sharks, and we've sort of developed a human equivalent that has similar properties. The long loop is quite key to bind into these GPCRs, as we call them, and our target is one of that class, and they're a very popular class for drugs. And so it's got functional activity. The lead candidate, I think, we've got evidence of efficacy in animal models of several different types of fibrosis. And I think it's the novel mechanism of action that we have here, I think, makes it a potential first-in-class therapy.
In the injection into the eye, presumably you would need to acylate the molecule?
We wouldn't need to, but we've looked at both the naked eye-body and the pasylated eye-body. So pasylation is a technology to extend the half-life. Because these are rather small, it means that the half-life is very quick in the mouse. So we can extend that by adding things to the end. And so you can extend the half-life quite considerably. You're right, we've got just as good data at the moment with them both, non-pasylated and non-half-life extended and naked.
I'm just thinking about viscosity.
Yeah. Because you do need, I mean, you know, these experiments, you've got to inject 1 microliter into a mouse, so it has to be high concentrations. So, yeah. Nothing else. Thanks, guys.